This video was developed with help from the Resources for Inclusive Education and Evolution workgroup. For more information and their other available educational tools, check the link in the description. Developmental plasticity permanently alters the phenotype of an individual after the initial exposition to an environmental cue. Because it permanently alters the development, measuring developmental plasticity is not as straightforward as measuring acclimation, and it usually requires a common environment experiment. Such experiment was done to investigate plasticity in adult body size of guppy fish in response to predation. The study examined guppies from two drainages in the Trinidad Island, the Yara drainage, situated close to the sea, and the Guanapo drainage, situated close to one of the most populated areas in the island. Two sites were sampled in each drainage, with and without predators. In the drainages, the body size of the fish varied between sites, with smaller fish being present in the sites with predators. At this stage, you can stop and think about the benefits of larger and smaller body size in different environments, and why does measurement of adult fish captured in their environments of origin cannot tell us if size differences are due to phenotypic plasticity or genetic differentiation. In general, one can assume that larger size at reproduction will confer larger fitness, as fish can produce more gametes. In the case of this guppy species, it has also been shown that in low predation localities, sexual selection via female mate preferences favors larger male body size. In high predation localities, however, it is advantageous for males to reproduce as soon as possible and thus to mature at smaller sizes because of high predation-related mortality. Moreover, predators can preferentially prey on larger guppies and thus select for smaller size. These selective pressures could result in differences in body size between guppies, but simply observing them in their natural habitat, we cannot deduce whether the differences are due to evolutionary changes, which are the result of modified allelic frequencies in the population, or to phenotypic plasticity, which does not affect the genetic composition of the population. To disentangle between genetic and plastic effects on fish size, a common environment experiment is required. Thus, adult, live fish were captured in the 4 drainages, brought to a lab, and their offspring was raised in 2 experimental environments, a control one and an environment where instead of putting predators that would eventually eat the experimental offspring, the researchers used a predator cue, which is simply water with excretions of the predator fish that were fed with guppies but in a different aquarium. Young guppy fish from all locations were placed in each of the treatments, left to reach their adult size, and then measured at maturity. This is what the results look like. Blue full lines are fish from ponds with predators, black dotted lines are fish from pond without predators. Looking at the results, think about these questions. Regarding how does predation affect body size? Predation decreases body size in both drainages and for fish that were collected in ponds with and without predators, as indicated by the negative slopes going from the control to the predator environment. Next, comparing fish from different ponds, we see that the population from the Yara drainage, fish in ponds with predators, are generally smaller than those in ponds without predators. This indicates that some genetic differentiation for fish size does exist. In the Guanapo drainage, where fish are raised without predators, those coming from ponds with predators are also smaller. However, the tendency is inversed in the environment with predators. This is a typical example of a genotype-environment interaction, which is covered in another course from REE2. We also have evidence that adult size is plastic in response to predator cues, with all populations showing decreased adult size in environment with predator cues, as indicated by the negative slopes of the lines. The most plastic response is observed for the population from the guanapo pond with predators, and the least plastic response from the population from the Yara pond with predators. In this example, because adult fish size is a developmental trait, once an individual has reached its adult size, it can no longer change. So it is possible that an individual that had predator cues early in its development would have small size as an adult, even if there are no longer predators in its environment. This is what we call developmental plasticity.